Welcome. To those of you in our audience just joining us, thank you, and we'll get started in just a moment. Welcome, everyone. Thanks so much for being with us today. I'm Michael Morella, Managing Editor at US News and World Report. Thanks for joining us for today's webinar, Will Genetics and Genomics Deliver Precision Health? Our panel will provide an in-depth look at how genetics and genomics are opening up exciting opportunities to treat certain diseases early, or to even ward them off entirely, while also offering new tools to identify the causes of rare disease, thereby opening the door to new treatments. Our experts will also discuss the challenges that lie ahead to fully realize the benefits for all patients. Now, today's session is the latest installment in our US News Healthcare of Tomorrow virtual event series, exploring critical issues facing hospital and healthcare leaders across the nation. You can learn more about the future programs and find links to previous videos and recap articles at usnews.com/healthcareoftomorrow. We are recording video of today's session and we'll be archiving that on usnews.com. We're also simulcasting on Facebook, LinkedIn, and YouTube. Now, I want to welcome today's panel, but before I do, one more note. If you have questions, we ask that you please type them into the Q&A feature in Zoom on your screen. Later on in the discussion, our moderator will try to give voice to some of these as we have time. So now I am pleased to welcome our panel. With us today are Dr. Robert Green, Professor of Medicine at Harvard Medical School and Director of the Genomes to People Research Program at Mass General Brigham, Ariadne Labs, and the Broad Institute. Dr. Andrew Cantor, Chief Medical Officer at Intelligent Medical Objects. Dr. Tomi Pastanen, Director of the Genomic Medicine Center at Children's Mercy Research Institute, Kansas City. And Dr. Consuelo Wilkins, Professor of Medicine and Senior Vice President of Health Equity and Inclusive Excellence at Vanderbilt University Medical Center. Thank you all and welcome. Now I'll turn the mic over to my colleague and our moderator, Liz Whitehead, Managing Editor with the US News Live Events team. Liz?
Thank you, Michael. And thanks so much to our distinguished panelists for joining us today. They'll be sharing some of the exciting advances in genomic medicine, as well as the challenges that lie ahead in realizing its potential to revolutionize precision healthcare. Tomi, if I can start with you, can you give us an overview of where we are now? How have improved tools and new technologies sped the work of genomic medicine in the past decade? And speaking generally, how is that translating into more applications of these discoveries at the clinical level for patients?
Thank you, Liz, and happy to be here representing Children's Mercy with my esteemed panelists. So like you said, the past decade has seen a tremendous increase in next generation sequencing utilization in the clinical place, particularly in the field that we work on in medical genetics to diagnose rare genetic disorders, but also in oncology on directing therapy for cancer. So last few years has seen even increases in the speed of development, especially through technologies that are becoming more accurate, higher throughput, lower cost, but most importantly, with the amount of genomic data that is out in the public domain that allows us to measure every patient genome against a large number of what we call normal controls. And this allows actually even computational interpretation of human genome sequences in a clinical fashion, a completely unheard of achievement in just the past couple of years ago. So, but we're not there yet. So we still have challenges in improving the diagnostic rate. And we at Children's Mercy, alongside with the rare disease research community, are working on to really provide diagnosis for all patients, not just to the minority as it is today, even for rare genetic diseases. So the three pillars of our approach at Children's Mercy are: first, use technologies that are continue to improve, and we have newer sequencing technologies that allow us to see the genome even in a higher resolution than the past decade of next generation sequencing has achieved. Secondly, what we use in this technology pillar is we also measure how the genome functions. So we can measure, for example, RNA being produced and combine that to analysis. And that's already commonplace in cancer genomics, is measure RNA in parallel with the DNA. The second pillar for rare disease and genetics of rare disease is data sharing and crowdsourcing data. So sharing data with other investigators, other clinical labs that are doing rare disease sequencing for those genomes that we were not able to solve. That's the key part, to find new genes. There's still probably thousands of disease genes and conditions that we don't know of because we just haven't mounted the body of data. And because these are so rare diseases, no one can do this by themselves. We need to pool data together. And the last part of our approach is really based on the generous philanthropy of our community to the Genomic Cancer for Kids program that I lead at Children's Mercy. And that allows us to provide this advanced genomic medicine sequencing to all patients at no cost to the patients. Because unfortunately, the medical decision makers as well as the reimbursement is not working yet for the most advanced tests and their timely application to rare disease patients. So we hope that our program, alongside with many other rare disease and genomics program in disease, will lead us to a state where the insurers understand that applying complete and comprehensive genome sequencing early is the right thing to do, not only for the families to cut down those long diagnostic odysseys, but also for the healthcare system at large, and actually will reduce cost over time. But this will still take a couple of years. So we forecast that we're a few years off from kind of the pinnacle for rare disease genomics, knowing all the disease genes and sequencing the full genome alongside with the function of the genome. But the hope is, like you say, provide new therapies, to provide new targets for therapies, and being able to build precision-driven disease cohorts based on the molecular diagnosis of every single patient. And I think that's a worthy goal for all of us to try for. So
there seem to be two pillars, Tommy, before I move on to Andy, then Consuelo, then Robert. We have indication-based testing, right? When you have a constellation of symptoms with, say, a child, you don't know what they have. And then you have screening, which is where can you make that distinction for us, and then we'll.
Yeah, so what we are focused on is really the indication-based testing, and we want to lower the bar for indication-based testing. So when a physician thinks that there's something odd with the patient, there's something not common, genomes should be done much earlier than they are instituted today, and they should be instituted not only in the specialty care but also in the primary care. So that's really where we work on. This will trickle down benefits to screening applications, because the more disease genomes we sequence, the better we understand every single mutation and gene defect in the genome that lends itself to be utilized in completely asymptomatic patients, pre-symptomatically, like in screening-based tests such as newborn sequencing.
Thank you. Andy, Tommy just tested on it on a big data challenge. If you have all of this data coming in, it's highly complex on, an individual's genome. You're trying to process millions of people's genome and then make this actionable on the clinical level. Can you just give us an idea, as somebody who's spent their career in clinical biomedical informatics, of how challenging this is to put together systems that clinicians can draw on to actually bring this to the room with the patient to help them?
It's a really important challenge, Liz, and thank you for inviting me. And so why IMO and why am I talking about this? I guess most people don't know of intelligent medical objects, but we are actually the terminology in almost every major electronic health record within the country. And so we're constantly trying to translate concepts from clinicians' minds into things that computers can understand and health systems can act on. So kind of the sharp end of the spear when it comes to trying to deal with this data in a way that is actually functional and actionable by both patients and providers. First of all, I kind of break down the the genomic information into sort of three big areas when I'm thinking about it. One is what we've just been talking about, which is really around, disease risk predictions, susceptibilities within the patient. There's another sort of important area, which is in pharmacogenomics, which is where patients' susceptibilities or genomic variability impacts their ability to respond to treatments, particularly certain medications and so on. And then the last area, which is really fascinating, which might be stretching the frontier a bit, which is the actual genome of the disease. So many cancers, for example, have their own genomes, and it's important to actually understand when we're treating those patients, particularly things like brain cancer and so on, which are the kinds of cancer cells that are in that patient at that time, and how do we treat them? And so each of those pose really big challenges, particularly when you're dealing with the data that's not even about the patient, but it's about the patient's tumor. So you can imagine that that can be a bit of a challenge. I think one of the things that we always have to keep in mind is that there is research, and there's our ability to generate big data, and there is the need to translate insights or interpretations of that data into actual workflow. And I think whereas the sequence information is incredibly voluminous and has incredibly high levels of specificity, most patients and most doctors don't think at that kind of level, right? They just care, is this patient at higher risk? Is this patient at low risk? Is this patient gonna, break down this chemotherapy drug faster than they're supposed to or not? And so part of the challenge is as we're kind of collecting all this data, one is ensuring that it's of the right sort of quality, and I'm hoping that our panelists will talk a little bit about ensuring accuracy and quality of that data, that it's representative of the kinds of patients that we really are interested in, and that that information can be transformed into some kind of actionable objects that both patients can understand from a risk understanding and a conversation with the clinicians. More and more of that's happening in the primary care settings even. But even more so when we start to build technology, or these electronic health records and health information systems continue to evolve, that they're starting to be able to act and support clinicians by sort of being a augmentation of their clinical practice, because no one's going to remember all the gene sequences. No one's going to remember all of the alleles and all the differences when it comes to taking care of patients. So I think that's context maybe as we go on for the rest of the session.
Oh, no, it's very helpful. Thank you. And Consuelo, I wonder if you can pick up on something Andy just touched on. You've been laser focused on equity issues in every point in the healthcare continuum at Vanderbilt. Can you talk about the challenge of applying genomic medicine to all population groups? There's some big issues there. Can you discuss what some of those are, and we can discuss later in the conversation how to address them?
Yes. Thanks for having me, Liz, and for the question. The idea that we can use genomics and precision medicine to really think about people as individuals and all of their experiences is something that excites me a lot. So, this approach to considering an individual and their variability is exciting for many reasons as it relates to equity, because a lot of what we talk about revolves around the idea that we are different and that we need to understand those differences. Unfortunately, when we look and we're thinking about it from the genomic standpoint, the information, the evidence that we have to guide us doesn't reflect the diversity of the populations that we're talking about. So while it may be beneficial to know and understand and think about, from the social context, the environmental interactions there, right now in the United States, more than 80% of the individuals represented in our large-scale genomic databases are people of European ancestry. So how can we actually use the evidence that's been generated from this population that doesn't reflect the diversity of the people who live in our country, the people that we're taking care of? It brings a lot of challenges. So we know that people who are of recent African ancestry have more genetic variability. So again, if we're thinking about what these genes or mutations, or if we're focused on pharmacogenomics, what are we missing? How can we actually take that evidence and apply it in a population that isn't fully represented? So that's one of the biggest challenges that we have. And then, of course, as we start to think about from an equity standpoint, how we would even implement genetic testing, whether it's for a specific indication or screening, when we have issues related to access to care, when right now having access to care or insurance in this country is tied to having a job. And then, being in an environment where you actually have access to the additional services that you might need to understand the genetic results. And then we could also, certainly talk about some of the historical challenges related to historic abuses and research that make people less willing to participate in or believe or uptake these newer technologies and medical innovations. So there are lots of challenges that we have to address related to equity pretty broadly.
No, that's great. And I'd actually like to come back to every single one of those as the conversation moves forward. Robert, I'm wondering if you can come in now. Tomi's discussed the importance of genomic research to help diagnose and treat rare diseases. Consuelo and Andy have sketched out some of the challenges. You're currently directing a very important initiative, the Baby Seek project, involving the genomic screening of newborns. Can you talk about that and maybe also discuss the difference between risk stratification and diagnosis, and maybe give us some real world situations and what you think the benefits of this project will be?
Sure, and thanks for having me as well. So just to take one step back, precision medicine is a buzz term that's in the title of this whole seminar. And it can mean many things, as you alluded to. It can mean personalized care at every level. It can mean wearables. It can mean, big data integration about an individual. Sometimes it can just mean tracking your labs in some sort of sensible way over months and years. But I do think genomics is often recognized as the tip of the spear for precision medicine. And that's because it has, in some cases, very, very powerful predictive capabilities. And what's ironic is that those cases have colored the entire perception of genomics so that we feel like we have to imbue it with all this pseudo accuracy, when in fact, it could be used in the same way as Andrew was talking about. Lipid levels are used, or lifestyle is used by physicians to talk about risk in a much more generalized way. Hey, you have an elevated cholesterol, you're at increased risk for heart disease and stroke. Hey, you're carrying a cancer mutation, you're at increased risk for these types of cancers. Now let's talk about what to do about it. And we get so hung up on the precision penetrance and the numbers that we can't see the nose in front of our face, which is we could be helping lots more people with genomics. So that's one thing. I'd like to reinforce the diagnostic uses of genomics and the risk stratification uses, as has been alluded to. Even in my own discipline, when we talk about sequencing healthy people, we slip into the language of diagnosis. Hey, we found a mutation, but it might not mean that we can diagnose cardiomyopathy. Well, it never meant that. All it meant is that you have a DNA change. Third, we've been talking about transforming research data into action, and I love that framing. That is, in some ways, the whole universe of implementation science, and that is the kind of science that we've been doing in our research program, which is taking things which have been discovered and trying to go that last mile to bring them to care with as much equity as possible. But obviously, there's always going to be early adopters, as there is with any technology, who want to try these things first, and other segments of society that hang back. And to some extent, the early adopters are taking chances on things where the benefit cost ratio and the risk has not been fully vetted as well. So in all that context, I've been working for about 15 years on the portion of genomics that has to do with sequencing ostensibly healthy people and how we could use that as one of the ways we could revolutionize our healthcare system to become less of a sick care system and more of a truly wellness-oriented system, a system that keeps you healthy. And we've done a lot of work with adults, and more recently, we've done the BabySeq project, which is the world's first project to whole genome sequence healthy newborn babies. Now, some people will say, it's amazing the reactions you get. Some people will say, well, of course, that's obviously where we ought to go. And other people will throw up their hands and say, this is the most ridiculous, dangerous thing we've ever seen. But we've done it. And in the small sample size, we found that 11% of ostensibly healthy babies are carrying disease-associated mutations, either dominant mutations or biallelic recessive mutations. And in some of those, we found evidence of the disease that wasn't present, that wasn't obvious any other way. So let me pause there because I've said a lot of introductory stuff, but we're very excited about what it might mean to bring a public health prism to population genomics and what it might mean to take that as early as possible in the lifespan.
Thank you. Consuelo, and then Tommy, and then Eddie. Can we go back, Consuelo, to screening and testing these different population groups? If we don't have reference data that is perfectly aligns with somebody's ancestry, does that change what we have to tell them in terms of what they can expect for the relevance of the results? How do you handle that question if somebody's being screened? I mean, do different ancestries, do they have different results where you have to have a couple gene anomalies in order to raise your level of disease susceptibility? I mean, has it changed the assessment of risk factors and what you tell the patient on the usefulness of the screen or test?
That might be complicated to answer just broadly, but let me maybe take a specific view, a piece of that, so, related to Robert's description of BabySeq, where they're doing whole genome sequencing in babies. Okay, so we don't necessarily need to have data that is specifically reflecting, the ancestry of the population. We just need to have it more representative than it is now. And because it's not, if we are doing whole genome sequencing and we're identifying specific mutations because they've already been identified in a population of people that's predominantly of European ancestry, and now we're doing this whole genome sequencing in babies who are of recent African ancestry, as an example, I imagine, and Robert can tell me if I'm wrong about this, but based on prior evidence, we're more likely to see variants of undetermined significance in that population, in those babies of recent African ancestry. So what does that mean? So, they may be variants that are important. They may be variants that indicate a risk of disease that we just haven't determined yet because we've not studied enough people of recent African ancestry to know if that makes, to know how to interpret that. And so if we're doing that, we need to be able to tell those parents, those families, that they're more likely to get these variants of undetermined significance. And it may be less predictive. Maybe the results may be less helpful for them. And so there are lots of other things to think about that related to that. For instance, if this were eventually sequencing that were offered at cost or that we'd have to pay for, if you have a variant of undetermined significance, hopefully we'll do better research in the future and we'll know what that means. But then, what's going to happen to that family? Is someone going to reach out and tell them we've learned something? Do they need to be tested again? What's the follow-up? So there are lots of unknowns related to that. And hopefully that was enough of a response to your broader question, which I think varies based on, the kind of genetic testing you're doing.
No, that was great. Thank you. And Tomi, can you pick up on this, because you have an even greater challenge because you're dealing with rare diseases. Can you pick up on Consuelo's point?
I think her points are exact on the mark. We're challenged with the recent African ancestry when we interpret clinical sequencing data. So if we look at, if I take you a concrete example of looking at rare genetic variants that alter protein coding sequences and compare African ancestry and Northern European ancestry samples that we get through our lab, we see 5 times more variation in African ancestry samples. That is rare. It's not truly rare because in their population it's probably relatively common, but because the reference populations are so driven by Northern European descent, we don't know their true prevalence. And most of our power in genetic testing for rare disease is driven by rarity of variation, and we don't have good enough spectrum in all the ethnicities, and this is a huge challenge. There hasn't been a study that I know of that would compare diagnostic accuracy for rare disease across different ethnic backgrounds. I think it's a study that we should do, and I'd be really motivated to work with others to truly figure out, because the problem of variants of unknown significance is like Consuelo said, huge among populations that have poorer reference data, such as African ancestry.
Thank you. Andy, can you pick up on some of these points? You've done work over the years in this area.
Yeah, so I think it's really interesting and absolutely appropriate, because when you look at the science, I think Robert talked about sort of implementation, and that there are certain examples where these genetic variants are pathognomonic, or in other words, almost 100% penetrated, cause the disease. But there's all these other situations where it's not really clear. And so we can do a lot of science on actually looking at the variants and looking at the proteins and modeling what that's likely to do. But we're getting these samples often from biobanks, which are sort of kind of taken away from the patient and the patient histories. And there's this kind of a real struggle to actually connect up the real world experience of people with these variants. And the National Academies of Science, Engineering, and Medicine, like literally yesterday, just released a big report on diversity in research and the need for ensuring that we're getting underrepresented samples into our research and clinical trials and others. And as we think about, like, how do we make that bridge, and how do we make sure that the data that we are associating these variants with, the life experiences of these patients, the diseases, their disease courses, what drugs worked, what didn't, those have to come from the health system of those patients, right? And in clinical trials is a very ivory tower sort of health system approach. And so you're getting a very narrow view of what that patient's experience is. But there's more effort today on bringing real world evidence and real world data to these questions. Can we take data actually out of the electronic health records and combine it with the genome so that we have a much more representative view of what these patients might experience? But then the challenge is who's in the EHRs, what's the quality of the data in those EHRs, and are we really bringing in the risk groups, both racial, ethnic, LGBTQ? There's lots of different groups that have different profiles, and so I think it's a little bit. We have to be a little humble when we're trying to leap from the individual rare disease gene to what that means in a population. And over time, that's going to get better, because as we become more inclusive, as we become more real world, hopefully we'll be able to validate the kinds of results that Tomi was referring to.
Robert, can you pick up on some of these points?
Yeah, I think that we should try to distinguish in our minds a little bit disparities that are inherent to the entire society at large and the entire healthcare system from things that are specific to genomics. And as Consuelo pointed out, I agree, some things are specific to genomics. The whole area of variants of uncertain significance, for example, absolutely true. Ironically, when you think about screening, we're probably thinking about a space that is a little bit more, a little bit less vulnerable to population ancestry differences in some ways. So for example, most people who are seriously entertaining screening with genomics, certainly we in the MedSeq project and the BabySeq project, are only talking about returning pathogenic or likely pathogenic variants. And they're only talking about disease gene validity categories that are definitive and strong. And of course, they're leaning into the clinical actionability for such things. So the search space, if you will, for preventive screening in genomics is much smaller and probably a little bit, not entirely, but a little bit less vulnerable to population differences than the diagnostic space, ironically. So that's my comment, I guess, about disparities for the moment. Yeah, let me pause there.
Thank you. We have an audience question, which we're going to turn to because it's important. You all in other conversations have raised the shortage of trained genomicists, geneticists, counselors, and the problem is a lot of people are coming, patients are coming in through primary care doctors who aren't trained in these areas. Tommy, can you start off and characterize where we are with the need for specialists in these areas, and what do the med schools need to be doing? I mean, do we have a quarter of what we need? Do we have any percentage of what we need right now? And how are we working with primary care doctors to make sure they have the resources they need when they're working with patients?
Sure. So I think everyone quotes deficiency of sufficient genetic counselors and medical geneticists in society to interpret large-scale sequencing to primary care providers. And we see it in our own health system as long queues to genetic specialty service. And this is a real problem, at least in our health system. But I believe it's a general problem. And it's a complicated problem because I think it goes into health economics and things that I don't understand on how hospital leaders decide on where they invest. But obviously, medical genetics has always been a relatively small specialty field, and I'm not entirely sure if the medical schools have increased training in genomics and genetics over the past years, because it's still considered a niche area. And a lot of the work we do is very computational understanding. It's kind of in between biology and computer science and statistics. And I think our medical training is not extremely well prepared to take the full advantage of genomic information to the primary care providers. But I think it's a complicated problem. We see it in practice as long queues to specialty care, and I think that's the experience from many other health systems as well. I think some of the work on kind of the front end of a genomic diagnosis, that the lab that I work in is alleviated by automation and higher throughput systems. We don't need as many people anymore to interpret the primary genetic data, but interpreting that to the primary care providers and physicians and what to do next, that's the gap that we have today.
Consuela, can you weigh in on this? I think you wear a couple hats, and also could you also, again, if you touch on the diversity issue, because there's obviously not been a great history with how people of color have been treated by the healthcare system. And if you're talking about data and privacy, there's a trust issue too. And so having people who look like you and have your experience, life experience, is important. So not just having trained geneticists, but also diverse geneticists and counselors, et cetera. Can you talk on all that?
I think you hit all the points. First I'll say that at Vanderbilt, just last week, we graduated our second class of Masters of Genetic Counseling graduates. So very exciting that, and most of them will stay in Nashville and be employed here at Vanderbilt. So, that's, speaks to the need for them. Of course, we still have challenges with this, this great resource of people who can actually explain well, these results. It's poorly reimbursed, if at all, in many cases. And so, if they are the right people to, or, among the best people to communicate these results, then we have to have ways of actually billing for services or being reimbursed under some model that allows us to support them. We certainly do know that there are too few people who are from minoritized backgrounds who are in all of medicine, health, science, and certainly genetic counselors as well. We have seen more recently pushes for individuals to be trained with, to do their work with more cultural humility, to understand some of the, historic abuses that might prevent or, provide a different context for people to understand their results. And I think there are some great programs that are evolving around the country that do that. It's not necessarily a substitute for that lived experience, but I think, given the demographics of the workforce, we have to embrace it as it's everyone's responsibility to be able to communicate the information effectively. I will say that, and I think Tommy mentioned this, that we just don't do this well in general as clinicians, as scientists, and that's communicating information, science, health information to the general public. And when we're talking about something as complex as genomic results, where we're talking about risk, often there's a component of that that's beyond literacy and more numeracy. And how do you actually, again, communicate this without having visuals available? I think we could do better in general with just how we make this information available to people, and lots to learn in that space.
Actually, I'm going to turn to Robert, but you worked on an interesting study that you were talking about. It's the literacy level that determines people's openness to getting involved with some of this testing. Was that the case? Can you talk about that, Consuelo?
Consuelo.
Yeah, Consuelo.
Yeah.
You did some research.
I thought you were asking Robert.
No.
Yes. So early on, when we were starting to prepare for the All of Us research program and launching it, so that's the 1 million people. The goal is 1 million people enrolled in this long-term study, precision medicine study. We wanted to try and understand: do people actually understand? Do they know these terms? Precision medicine, genetic testing, pharmacogenomics. There's a lot of variability in their genomic literacy or genomic health literacy. And there are differences based on race, but also more on educational attainment. So the starting point for many people is, with their literacy level. We didn't assess their numeracy in that particular study. But what we've also learned as part of the All of Us research program, when we ask people, what information, who's going to help them interpret their results, there are, small groups of people who say they have no one who will help them interpret the results. There's some people who say they're going to go to the emergency room to get help with their results. People just don't know what they're going to do or how they're going to understand this information. So we need to have a better system, again, of supporting people in their journey of understanding. And we also have to respect that in many cultures, and depending on your educational attainment and resources, you're probably having family members or friends to help you understand that. And so people with limited resources or access to resources are going to need more support in that.
Thank you. So Robert, you have several really important initiatives that pull together a lot of these issues. how people are counseled, the trained personnel that you have advising the people participating, the families. So can you weigh in on a number of these points?
Well, sure. I mean, imagine if we only allowed a nuclear physicists to use the information that came out of X-rays, and we only allowed PhD chemists to interpret the information that came out of blood chemistries, and so on and so forth. It would be ridiculous. You couldn't practice medicine. And yet that's sort of what we backed ourselves into a corner with in genetics, with this whole concept of genetic exceptionalism. You know, the superb care that medical geneticists and genetic counselors do provide probably has some rationale in certain conditions, maybe reproductive conditions where terrifying decisions have to be made under short timelines. But in the rest, where genomics is just medicine, I think we really need to reframe it as medicine. And so that's what we've tried to do in MedSeq and to some degree BabySeq. Now, genomics is complex, but you can train primary care. We demonstrated this in the MedSeq project 10 years ago. You can train primary care doctors to read the results of a whole genome sequence and respond appropriately. That's pretty startling. If you get into sequencing newborns, you're going to be uncovering risk of really esoteric rare diseases. Probably they're going to need more help on that. But, you know, we've managed to make this work in a lot of different scenarios. And I don't think we should be intimidated by saying, you know, oh, it can't work. I think it can. The other thing I would mention is just in full transparency, I co-founded a company in this space, but there are companies that are using telemedicine genomics expertise, either medical geneticists or genetic counselors who are hired to offer this, who can distribute esoteric knowledge much more efficiently, much more readily, much more inexpensively than the academic-based concentration of genetic experts that we find ourselves in with physical care. So telemedicine, which of course has gotten all sorts of new life with COVID, is really, I think, going to accelerate the implementation of genomic medicine for those reasons.
Thank you. Andy, have a follow-up question again for the poor primary care doctors who are trying to plug into the system here. Can you talk about what kinds of assistance we need to tie these stakeholders together? You know, are there decision trees that we could give primary care doctors to help them understand when they get a patient screened, or you know, indication-based testing for something, what they're supposed to do next?
Yeah, I think that's a really important point, Liz, because I think, as you know, Robert was pointing out, is that, you know, ultimately for most people, they're likely to interact with genomic medicine outside of these really crisis situations. And I think, you know, there's something like half of all primary care doctors are prescribing medications that, for example, would be impacted by a gene variant. And, you know, 1 in 5 of those prescriptions currently being, you know, given out to primary care patients could be potentially altered based on an understanding of that patient's, you know, genetic variation. And so we really do need to think much more broadly about how to incorporate this kind of information into primary care. And I can think of it on sort of maybe 2 directions. The first is kind of on the inbound side, which is like, how do we train physicians to do what they kind of did in medical school, hopefully, which was really understand the patient context, the family history. Are they capturing the context of these patients well enough to inform the, you know, positive predictive values of some of these tests, or you know, or are they simply just running a test and getting the result and trying to come back? I think that we really want to try to bring more data in when we're making those decisions. And then as this results come back, I mean, Robert, I think it's great. And I'd love to know a little bit more about how much time it took them to be able to actually understand a full genomic sequence and turn it into actionable advice to a patient. But most, I would think, primary care physicians are complaining about a single additional click on their screen, let alone the interpretation of a block of text, which could be fairly extensive. So I think people are hoping that the technology will simplify this somehow by extrapolating or transforming the kind of the voluminous data into something that is more rapidly consumed and then transferred. And then obviously when patients aren't getting what they want, that there should be an escalation to someone who can answer those questions. But trying to empower physicians at the front point of care who might be scared to death, as Robert said, they've got this weird assumption about how dangerous this kind of conversation actually is, which is probably in most cases not true. So I think there is hope that we can do that, but we're not yet in a place where technology is able to do that. We're still in the raw sort of hypothesis generation and dealing with lots of diversity not in patients, but in sort of raw data. And so I think we've got a little bit of ways to go before technology can really help that primary care physician, do that. Although I do believe there's a lot of good work being done on data visualizations, as Consuelo was saying. I mean, a lot more about risk communication, and we can be better providers by using those.
I'd like to move in a second to how we determine what to screen for, but I have one follow-up for Tommy. How big an issue are privacy concerns? For example, when you're talking about trying to get different institutions to share their data, but you're dealing with rare diseases. You talked about crowdsourcing. What are the obstacles that people are raising to do that?
So I think this is a great topic. And in terms of rare disease, what we're dealing with mostly are families that have looked for a diagnosis for years. They're extremely motivated to do whatever it takes to understand what's wrong with their child. So there's a risk stratification based on the potential benefit of data sharing. And I think Robert can follow up on this, but we are at the kind of top of the benefit risk ratio in data sharing when we deal with rare disease and unsolved rare disease and sharing that data. Families are motivated, that's what they want to do, not even knowing that it's probably not going to help their own child immediately, but knowing that it may help other families in the same situation in the future. So we are in a good situation with rare disease, with severe childhood onset disease, where people actually want to share. The community understand that we need to share to find the answers. But of course, then talking about genetic risk and going down on severity of disease and probabilities of disease, the benefit risk ratio becomes different in data sharing. But I think we should be realistic about what is the breach of genomic privacy, what is the likelihood of breach of genomic privacy, and how much information do families share through social media and other instances that is probably more harmful than having your nucleotides in some data space. And typically the data sharing is it's always, it doesn't have any HIPAA information. It doesn't have, it's anonymized. We do attach some standardized medical terminology to symptoms and then the DNA sequence. It takes a lot of work to work up who the patient is and breach the genomic privacy. So one should always scale the risk benefit ratio. And I think the risks are relatively low.
Robert, feel free to respond to that. But in addition, could you talk about how the decisions are made, for example, during newborn screening, what to screen for, and how over time that would be modified, and what stakeholders have to be involved in deciding what you're screening for, whether it's newborns or adults?
Yeah, thank you. I agree with Tommy very much that we have over-indexed on legitimate concerns about psychological distress, privacy, and discrimination. Those are absolutely legitimate concerns. We should never be cavalier about them. But the data that we've gathered so far, including material that we've done sequencing healthy adults and healthy babies, suggests that, number one, people who volunteer for these testing, for these are well equipped to manage the results, even when they learn risky things. Number two, the number of times that you actually get insurance companies underwriting or breaches of public trust or databases is vanishingly small. Now you compare that against all of the information, including life-saving information that is present in a genome. In our work, we believe now that based on our comprehensive sequencing of apparently healthy adults and children, that between 10 and 20% of all of us walking around have a dominant disease-associated mutation. Over 90% of us are carrying recessive mutations that may be very influential in terms of our children's and grandchildren's future health. 80% of us have an atypical response to at least one family category of medications, and well over 50% of us are in the highest levels of risk in terms of a polygenic risk score, which of course may only tell us about the genetic component of that risk, but is still generally agreed to be useful at this point. Now, this is important life-saving stuff. So it's really not a question of, it's really a question of logistics. How do we get this into the hands of people who know how to act on it in a reasonable way? We should be able to do that. So I agree with Tommy.
That's well. Feel free to pick up on the points that are made. And I also, in our last minutes in the webinar, I'd like to discuss the importance of cascade testing, family testing. So do you want to address either or both the previous points and this point?
Maybe I'll do the previous points in that, I do think that the privacy risk, the risk of harm might be small, but the perception of those risks are highly variable across populations. So when you're talking about people who are from groups that have been previously harmed and abused in research and medicine, then their belief that their privacy might be violated or that some harm might come to them, that there's some economic fallout, that their DNA could be used in the criminal justice system. Like all of those things are amplified in those populations because these are things that have happened to them before. So I think, we have to recognize and not discount that. And we also do need to make sure that we are understanding when we're talking about these minoritized populations, that there is perhaps more of a risk for re-identification. If they're only less than 15% of the database is people from a certain background, and then you start adding in other information about them, the risk for people in those smaller groups is potentially higher. So I do think we don't want to ignore that. We need to acknowledge that. And that gets into the issue of trust, which is the most important thing that I tell people we need to think about when it relates to trust is the transparency piece. Like being transparent, like, yes, validating, like you have a real reason to be concerned, but this is what we're going to do, and these are the oversights, and reinforcing that I think is really critical.
Thank you. Andy?
Yeah, I think actually it's hard to go after with that, Consuelo, because I think that really needs to take precedence because we tend to be as a clinician and sort of a scientist, is a kind of an arrogance about well, this has got to be good for the patient, and it should outweigh even Robert's statistics. I mean, almost everyone's going to benefit, right? But there's these sociocultural contexts that we can not avoid here. I do think that there is some importance in being able to kind of demonstrate that value proposition to people as they're going in, and that whether it can be shown in some real world experience of their antihypertensive drug, or whatever, that they may be having certain symptoms or side effects based on us not understanding why they're reacting to a particular way. I think we need to do a better job of sharing those experiences with people. And frankly, I don't know, Robert, you're overseas right now, and Tomi, you come from there, but into the European sort of understanding where the legal regulatory protections really need to be made very clear early on before we open the floodgates. I'm not as well voiced, but certainly with the GDPR on the privacy kind of front, there's evidence that we can maybe set up some stronger guardrails to help protect people from perceived or real risks.
Robert, I'm going to ask for final comments from all the panel. I do want to get your thoughts on cascade testing and family, whether, when you have screening and you turn up these conditions, do you need to counsel the family that everybody should be screened, and how does that get handled, paid for?
Sure. And as I approach that, Consuela, I would never mean to minimize the topic. And I think you're exactly right that it's perception. But it is a struggle to change perception. And I'm not sure how to do that. And whether it's perceptions or it's fears in the general population of discrimination or whatever it is, in my view, some of these perceptions across the population have been holding the entire field hostage, so that the entire field can't move forward. And it's a catch-22, because then the field can't demonstrate all the benefits. It can't save the babies. It can't save the women, including women of color. It can't, if the field can't populate itself, it can't move forward. So risk is, real risk is real, perceived risk is real, but somehow we have to figure out guardrails that allow us to proceed. One of the areas is that I think is incredibly underutilized is cascade testing. When you do find someone who's carrying a dangerous mutation, you know that unlike the rest of the population, their first-degree relatives have a 50% risk of carrying that same mutation. This is prevention on a platter. This is the ability to hand somebody a cost-effective screening tool where the prior probability is 50%. So I think it's a huge failure of imagination that we've not been able to understand that genetics affects families in such a way, and we can literally save family members' lives by pursuing cascade testing more prominently.
Thank you. So we'll do 3 minutes for our lightning round to finish up. Consuela, your final comments?
So this has been a great discussion. I've enjoyed participating and also learning. I would push for one thing, as we've talked about the issues related to inequities, the lack of diversity in the samples, the databases, the research studies, and certainly as also among the workforce. If we're going to do good science, and I think Robert alluded to this, we feel like we're stuck, right? We have to at some point stop trying to just add these minoritized and historically excluded people into research that's already going. And we need to take some time to actually build programs that are in some ways restoring or repairing these harms that happened in the past with excluding people or not including them. So we need like a huge influx of dollars, resources, people to bring in some decent number of people from these other ancestries into the work. It's, in my mind, unacceptable that 60% of the world's population is of Asian descent and 17, 18% of recent African descent, but our databases don't reflect that. If we're doing good science, we have to work towards that.
Thank you. Tomi, Andy, and then Robert, final thoughts? Tomi?
So I think it's been a great discussion, and for me, I have to talk for the families and the patient advocacy groups. The families are looking for answers. They want the most comprehensive test, and the insurers don't see the benefits the same way as families that are in search for answers. So actionability in medicine is very different for a rare disease family than it is for an insurance company. And we have to build that bridge for the payers to understand that there's a tremendous human and financial cost for not having the answer for years and going through futile testing and futile clinic visits. So I think that's the gap that I'd want to address at the end.
Thank you. Andy?
Yeah, I think from my perspective, we learned a lot from the pandemic in that we don't handle big data very well, and that the quality of that data to actually help us decide and make actionable choices is not where we usually think it needs to be. And I think of this, even before this genomic sequencing came with some of the 21st Century Cures Act and some of this desire for interoperability, there's going to be a complete tidal wave or tsunami of data that falls on patients and on providers. And if we aren't very thoughtful about how to manage that data, it will swamp and drown us rather than really empower us to improve the lives of our patients and our own and our societies. So I think we need to be very thoughtful about what kinds of tools we can do to transform, ensure the data is the right quality, and help empower patients and providers to act on that data.
Thank you. Robert?
3 quick points. One, I think most of everybody who's listening probably understands this, but in case there's any doubt, array-based testing like 23andMe and Ancestry is not sequencing. We are really talking about the new era of sequencing. It's 10,000 times more granular and much more relevant to medical. Number 2, I believe we need to right-size our risk-benefit understanding of all of genomics, which has been skewed and over-indexed on risks that turn out to not be empirically supported. And number 3, as we and others are showing, whether it's in the newborn space or adult space, there is so much actionable, treatable benefit. Over 800 treatable rare diseases now, where the recommended uniform screening tests for 30 to 60. So we, as Andy said, we have a tsunami of actionable, life-saving information that's available to us. We're long past due to bring it into the hands of our doctors and our patients for better health.
I'd like to thank all of you for a wonderful conversation. Doctors Consuelo Wilkins, Andy Cantor, Robert Green, and Toby Passanen. I think that genomic medicine has tremendous potential for all of us. There are challenges ahead, but we are fortunate to have experts like you who are going to help us meet them. Thank you.
Thank you all.